Tumor mutation burden is one of the new biomarkers that we're looking at right now. The CheckMate 227 trial was looking at ipi-nivo versus chemotherapy and chemotherapy plus nivolumab. Now, this was a very complex trial, and so they utilized a biomarker, tumor mutation burden, which is essentially the number of mutations per megabase, and they used an assay called Foundation 1 in the tumor tissue. And what they discovered is that patients who had high TMB, tumor mutation burden, greater than equal to 10 mutations per megabase was how they defined it. These patients had a higher progression-free survival with the use of immunotherapy, specifically ipi-nivo, ipilimumab and nivolumab, compared to those patients who had low TMB or less than 10 mutations per megabase. Now, the patients who received immunotherapy, who had high TMB, did much better for both progression-free and overall survival when compared to chemotherapy. Now, this is not an accepted standard of care yet, because there are a lot of questions about what should the cutoff be for tumor mutation burden and what assay should we use. Remember, the CheckMate 227 study used the Foundation 1 assay. This is a platform that changes, and so other companies may have different cutoffs. So all of this remains to be validated and approved before we can use it for prime time. Ipilimumab and nivolumab is a very well-tolerated regimen. We've given it oftentimes in other thoracic malignancies beyond just non-small cell lung cancer, for example, in small cell and in mesothelioma. So we do know that it can be safe. It can be easy to give. There are patients who tolerate it very well. But it's important to note that immunotherapies do carry toxicities. And so we do have to watch out for colitis, fatigue. There are also any autoimmune event, such as any inflammation anywhere in the body, is usually what I tell our patients, is always a risk. But generally, patients do feel pretty good while they're on this regimen. And it's certainly, though, important to educate our patients about the toxicities that can occur.
There are actually multiple drugs in this space that are either targeting PD-1 or PD-L1. Targeting PD-1, we have nivolumab and pembrolizumab. PD-L1, atezolizumab and durvalumab. Atezolizumab is also approved in lung cancer, in combination with carboplatin, Taxol, and bevacizumab in the frontline setting. That's based on the results of Empower 150. In Empower 150, a little more complicated because you have 4 drugs versus 3 drugs. So you have carboplatin, paclitaxel, bevacizumab, standard of care, all for non-squamous, versus carboplatin, paclitaxel, bevacizumab, and atezolizumab. And that had a hazard ratio in a very significant range, so that's approved. The question is that any better than using chemotherapy with pembrolizumab and not using bevacizumab? Hard to tell because we don't have a direct comparison. Bevacizumab is a good drug. It improves chemotherapy. How much difference it's making here, I think still needs to be sort of sorted out. It does have, additional side effects that it adds. I would use this regimen, though probably I would reserve it for the patients who have EGFR mutations and have failed targeted therapy. Why? Because in the trial they actually allowed for that to be included. So there are some patients in there, albeit fewer than 10%. That's not in the U.S. label, to be fully upfront, though I believe they have approval for that in Europe. So at this point now, I would probably, if I had an EGFR mutant patient who I was going to put on their first immunotherapy, I'd probably use the Empower 150 regimen.
Empower 131 was a trial specifically evaluating the role of atezolizumab in combination with platinum doublet chemotherapy, specifically for advanced squamous cell patients. This was a very large trial. It was a 3-arm trial comparing carboplatin nab-paclitaxel to carboplatin nab-paclitaxel plus atezo versus carboplatin paclitaxel plus atezo. And really the primary analysis was looking at arm B and C, which was carbo-nab-pac versus carbo-nab-pac plus atezolizumab, specifically in a squamous cell patient population. And in this trial, we saw that the addition of atezo to carbo-nab-pac improved progression-free survival when compared to carbo-nab-pac alone. Importantly, however, we didn't see an overall survival advantage at the time of the presentation. And so I think this is certainly an interesting trial. We have another trial in squamous cell that did show a survival advantage with a very similar design, but with a different immunotherapy. So while it was good to see the progression-free survival advantage in this trial, atezo added to carbo-nab-pac when compared to carbo-nab-pac alone didn't translate into an overall survival.